Hello, my name is Ryan Profnick, and unfortunately Dr. Kaufman couldn't make it here today, so I'm going to be doing the presentation for her. She is affiliated with Vanderbilt University. I am an employee of Cook Myocyte, who is the sponsor of this study. So thank you, ICS, for allowing us to speak today. This presentation is slightly different than maybe what is traditional in a sense. We're not going to talk about the data per se, as that was already published a few times earlier this year. But what I wanted to do is talk about the very important lessons that we learned through the developmental program of this technology. For those that are unfamiliar, the technology, it's called AMDC, or autologous muscle-derived cells. And how this works is a patient gets a small muscle biopsy from her thigh, and that sample is sent to the manufacturing facility where a select population of muscle cells are isolated, grown, and then they're cryopreserved, and after quality release sent back to the physician, where in the clinic setting, that sample is thawed, diluted with saline, and injected directly into the urethral sphincter. The phase 2 program, which has consisted of 4 clinical studies, have tested a range of doses from 1 million cells all the way up to 200 million cells. And what we could say about this phase 2 program is it had an extremely favorable safety program. It also narrowed in on a dose that provided, at least suggested provided a reduction in SUI symptoms. And again, what I want to talk about today is throughout the almost 2 decades now of this clinical research, both the preclinical and the non-clinical, we learned, as you can imagine, a numerous amount of lessons, but there was no better area for learning lessons than the initial randomized control study that was performed. Now this program, which has safely treated well over 500 patients, is still moving through those phase 3 studies. So the endpoint. When we started this, we looked at the endpoints that were used in the device regulations, because that was really what was out there. So we looked at the 50% reduction in incontinence episode frequency. We also, throughout the program, looked at pad weight, both in a 24-hour and also the 1-hour pad weight. So when we designed the study, the concept was a patient was considered to be a responder if they met the criterion for any one of these. So what we found, though, unfortunately, was that 80%, when using this responder rate, when using this composite endpoint, there was more than an 80% responder rate for both the product and the placebo. So clearly that composite endpoint was too liberal. We also found that the pad weight test seemed to be confounded, and it was actually the IEF reduction that correlated with the improvements in quality of life. And then we looked at just the 50%. When we did a post hoc analysis, we looked at more stringent criteria and looked at at least a 75% reduction, as well as going down to less than 1 stress leak over 3 days. And what we found is, as we moved to the more stringent criteria, there's an even more positive correlation with IEF reduction, as one might expect. Now the placebo rate, again, we designed the study, and there wasn't really much data out there on what a placebo rate should be, especially for slightly more invasive technology. So we used power calculations of 30% responder rate, and again, what we found was actually the placebo rate was much, much higher with this composite endpoint than to be expected. However, again, when you just looked at the IEF reduction alone, there was quite a dramatic decrease in the placebo rate. So again, moving forward and trying to continuously build a program, the impact on the future study designs was we had to look at the placebo rate not just in general, but we had to look at it to adjust it for the technology, the study duration, and also the use of the IEF reduction endpoints. Finally, the third lesson that we want to talk about today is not all patients are going to benefit from this type of therapy. When we did this initial RCT trial, we did not exclude for hypermobility, and it was not part of the selection criteria. But that got away from the mechanism of action that was learned through the preclinical program in the animal studies, which in fact is to augment sphincter function. And while this isn't a direct comparison, a patient population that was interesting that we looked at was patients that had prior continence surgery. And when we looked at those patients, not only was the technology very safe for them, there was a marked decrease in IEF over the 3 days when you looked at the cell product over placebo. So the impact on future studies now is to look to exclude patients with notable urethral hypomobility, and then also look at this with a little bit more detail, this patient population that had prior incontinence surgery that did not get the results that they were hoping for. So finally, in conclusion, again, looking at the placebo effect, we know that it's most likely influenced by many variables. You have to look at the invasiveness of the treatment. Again, patients are coming in for a biopsy. They're waiting up to 2 months for their product to be returned to them, and then they're going through the injection and then 12 months follow-up for the primary endpoint and another 12 months for the secondary endpoint. So it's a long duration, and then also managing patient expectations. The outcome measures should be viewed in terms of their sensitivity to placebo effects, their clinical meaningfulness, and of course lining up with regulatory agency expectations. The primary data suggests that patients with minimal urethral hypermobility may actually represent the ideal candidates for this type of therapy. And finally, lessons learned from this first clinical randomized control study certainly strengthen the design and scientific rigor of current and future studies. Thank you.
Thank you. Any questions from the floor? It's the common experience with stem cell studies that the actual cells don't do the job, but the activation through the manipulation. So this is definitely worth considering when you inject placebo cells.
So I have 2 questions. One's for the audience. Why don't you guys ask any questions? These are really interesting papers. This society was founded on the concept of an interaction between research and the speakers and the audience, and that's how new ideas come along. So if you guys don't ask questions, I'm going to get up every time, and I can get very boring. My wife can attest to that. I think I congratulate you two for doing this kind of analysis. I'd like to just comment, it's more of a comment than a question, on the utility of composite scores, but in my judgment, the problem with most competent scores is that they're a yes/no. You make the 50% or you don't, the pad test or not. If you use a graded outcome for each score, a numerical score from 1 to 10 or 0 to 4 or something, and then you add the numbers together, I think you'll get a much more meaningful response. And then I do have a specific question though about the pad test. We find the pad test very, very useful and very, very reliable, and it's been published. I'm talking about a 24-hour pad test. But again, there isn't always a correlation. Many patients with stress incontinence don't experience the feeling of leakage when they leak and just find their pads wet. So if you just rely on a pad weight and a person feels it and their main complaint is the moment they're wetting and that it's embarrassing, then you'll lose all of those. And if you just rely on the IEF, the incontinence episode frequency, you'll miss all of those that are bothered by stress incontinence that don't feel the moment that it happens.
Yeah, thank you. So, certainly I'm not a physician, so I'll do my best to comment on that. From our perspective, a lot of this is about patient education when they're actually filling out the diaries, and the patient-reported outcomes are a really tough way to go. So it would be great if we found that correlation with the pad weight. I think probably the challenge that we have is discerning the difference between stress incontinence and overactive incontinence, and and seeing where the pad weights come. So when we look at the diaries, they're actually designed to try to tease that out. So maybe that's where the differences came.
Since your focus is shifting towards the treatment failures, in your future studies, would you be including an arm that focuses on a comparison with bulking agents? Since your product is so complex and expensive, I would think that you might want to compare efficacies with the current treatments for surgical failures.
Yeah, so there's certainly not enough time up here to talk about all the things that we've learned over the years, but one of the challenges that we have is if you want to run a randomized, double-blind, placebo-controlled study, you can't do something like that because you can't give a doctor the bulking agent and say, close your eyes and inject and hopefully hit the target. So we've struggled with that, and trying to line up with regulatory expectations. This is the path that we went to, but it certainly would be nice to have that comparison data.
Thank you very much.
Thank you.